Um,
But now let me see if I can give you host and see, hopefully it'll continue.
okay. You're asking me about technology?
Yep, so you're the host and it appears to be recording still. So you can, when you feel like sharing your screen, we'll see it here online and on our big screen, like a movie theater.
Can you see it now?
Sort of. I mean, I need to move it over to a different screen over here. So 1 second. I can see it, but they can't. So let me see if I can. I felt like asking Abby, can you come over here? So let's see. Do I hit? Oh, so how do I switch screens? Let's see. Should usually pops up right over here.
Can you just drag it from like the actual screen over?
Well, he's the host now. Can you hit screen 2 on top? Does it show screen 2?
You want me to stop sharing or it's okay?
Go ahead and stop sharing just for a minute.
Okay. Let me get water.
Okay. So how do I move these over? I'm gonna move this, I'll just move that over. Okay, now let me see if this works. Now, could you share your screen now? I think this will work.
That's weird. I have now multiple screens. Okay, let me try this one.
Okay, well, I think this, so now we can see your presentation without, and then you just have to put it into presentation mode. That's it right there. And so now we can see everything.
What do you see now?
Well, I can definitely, all, and everybody in the audience can actually see your PowerPoint slide now.
Okay.
And then I can still see you on one of the computer screens, but I think everybody will see you later when we get to the questions and answers. So this will work, I think. Everybody agree? Anybody else want a piece of pizza or anything before we get started? All right, Cesar, I think everybody can hear okay. Obviously, if you're in my class, please take some notes. If you're not, keep enjoying your pizza. Or it'll be recorded online. So, all right, Dr. Rodriguez Sanyono.
Oh my goodness. You're saying it like differently every time you say it.
I've only known you for what? How many years have I known you?
A lot. 25?
Is it? No way. Could be.
No, maybe 2000, maybe like 20, more than 20.
No. Yeah, so it's been about 20 years. Dr. Rodriguez, and how am I going to say it this time? Sayona. Did I say it right that time? Like I said, I've only known you for 20 something years.
I think that's the closest.
I can say for one thing for sure, he was a bit of I would say you're a bit of a bigger brother mentor type for many years in science. You know, you were several years ahead of me in your career. And so.
Not that many.
Well, a few years. I mean, I'm a lot younger than you, right? And so anyway, you're definitely a motivational person. I think a lot of people, you know, you're very popular among a lot of people. And so, you know, obviously we always enjoyed talking science and got to do a collaboration together, which we looked at caterpillars and aphids, and many people in my class know about it. Remember, caterpillars are these big chewing insects in comparison to aphids and piercing, sucking mouth parts, and they alter plant defenses differently. So this was the PI, or the main person, the first author on that publication that I was a collaborator with. And so he was actually at University of Toronto as a postdoc at the time, right?
That's correct.
And so we've also talked a little bit about Jennifer Thaler, who was your postdoc advisor. And of course, no plant-animal interaction class is complete without talking about Honorog. So they got to learn about Honorog and milkweed a little bit too. So, and of course we talked about Mayberenbaum and Eowulf and a few other people like that. So you're up there with those great scientists.
Wow.
So anyway, thank you so much for presenting. It's been a long time since you've been on campus. It looks about the same, but with fewer students.
Yeah, I think it has been like 20 years. I think I went there like 2003.
Yeah.
But I went back, I think.
Yeah, you did.
I gave a lecture for your class.
That's right. And I think it might have been similar too.
Yeah, it might have been 2007, 2008.
Yeah, that's right. Well, anyhow, so you're an ag, your official title is Extension Specialist, is that correct?
Yeah, that's the official one.
Could you tell everybody a little bit about what you do as an Extension Specialist, and then I'll leave you alone. I mentioned it to the students.
I can talk about it after the lecture.
Okay, sounds good. All right, well, without any further ado, thanks, Cesar, for coming here. And the title of your presentation is Tritrophic Interactions in Agricultural Systems.
Okay, thank you for having me and for this opportunity to talk about my research. This is, I'm trying to like summarize about 20 years of data, looking at how to manipulate natural enemy behavior in agroecosystems and how to improve conservation biological control. So just to start with a little bit about my background, where I got here. So I started my journey in entomology as an undergraduate student in biology at the Universidad Nacional Agraria. That's in Lima, Peru. There I studied parasitism of a newly invasive pest at the time, the whitefly Mysia tabasi in sweet potato. So after I completed my bachelor's degree, I moved to Oregon State University for my master's in entomology, where I worked on improving biological control through selection for predators that had shorter developmental times. So after my master's, I went to UC Riverside to complete my PhD, where I looked at the roles of specialized defensive cells that are present in avocados against herbivores. So after that, I moved to Arizona, to Phoenix, where I did my first postdoc at the USDA ARS lab. That was the Cotton Research Lab. Now they have moved to Maricopa, but at that time was in Phoenix. And this is where I learned how to collect plant volatiles. And I studied also the role of the volatiles from cotton plants on ligas and natural enemy interactions. So Dr. Meltzer, he mentioned that I worked with Jennifer Thaler. So after I completed my postdoc in Arizona, I moved to Toronto, Canada, where I worked at the University of Toronto. And there I was interested in looking at how these tritrophic interactions, the interactions between the plants, the herbivores, and natural enemies, how the importance of these interactions in natural systems. So I manipulated plants, as you can see here in the picture, where I damaged a single plant within a patch, or I damaged all the plants within a patch, or I left the patch of 3 plants undamaged. And then I looked at the number of herbivores and natural enemies on each of the plants to look at how a neighborhood influence these interactions. So whether you're near an induced plant or an uninduced plant, how that would affect the number of herbivores and natural enemies that would attack a plant. So after that, I moved back to the U.S. And I did my the last postdoc that I conducted. I moved to Michigan State University, where I worked. I did some work on the role of volatiles to attract the emerald ash borer. At the time, the emerald ash borer was recently found invasive pest, and it was moving quickly throughout the state, and now it's present in a lot of other states, including New Jersey. So at the time, we were looking at ways to find attractants for these pests to better monitor this insect. So here at Rutgers, I'm the blueberry and cranberry entomologist. So since I joined Rutgers University, my research program has asked both basic and applied questions. So within the basic questions, I have been asking, does domestication affect plant defenses against herbivores? Also, we have been asking, does pathogen infection affect plant-insect interactions? And on the more applied side, we have asked, can we use semiochemicals to manipulate insect behavior and reduce pest populations? So we are testing attractants and repellents for insect pests. And we are using them to develop different ways to manipulate insect behavior to control these pests. And we are also looking at attractants and food sources for conserving natural enemies of the herbivores. And today, what I'm going to talk about is the work that we have been doing, both to look at how domestication affects tritrophic interactions, could affect plant herbivore interactions, and also how we can use the knowledge that we have especially with plant volatiles, to manipulate the natural enemies and also provide them with food sources. So my research, what I'm passionate about is on tritrophic interactions. So these are interactions among plants, the herbivores that feed on the plants, and the natural enemies of the herbivores. So earlier on, researchers in host plant resistance, they look at different traits in the plants that are important in defense against herbivores, while the researchers on biological control, they studied the impact of natural enemies on the herbivores. And these two methods were viewed early on as independent strategies. However, this view changed about 40 years ago when Peter Price and his colleagues wrote a seminal paper arguing that the theory on insect-plant interactions cannot progress realistically without considering the third trophic level. So, since then, there have been a lot of studies looking at these interactions. However, most of these studies looked at a single plant species, a single herbivore, and a single species of natural enemy. So early in my career, I questioned whether these studies were realistic, especially considering that plants are not always alone, and plants are challenged by multiple herbivores, and also you have multiple natural enemies that can attack those herbivores. So I'm also looking at what are the ecological factors that influence these interactions. And more recently, we're looking at whether these interactions can be exploited in agro ecosystems. So I will start with a brief review of plant defenses. As you know, plants cannot run away from their enemies, but instead they employ several strategies that reduce the number of herbivores and the amount of feeding damage on the plants. So they can produce toxins, anti-feeding compounds, and also growth inhibitory compounds. And some of these are stored in the specialized structures like trichomes. So these compounds reduce the growth and decrease the feeding of the herbivores on the plants. In addition, plants also produce volatiles that attract natural enemies of the herbivores. And these volatiles may provide an indirect protection to plants when the natural enemies of the herbivores are present. So defenses in plants against these herbivores can be induced by herbivore feeding damage. So one such type of defense of inducible defense are these proteinase inhibitors. So these, as the name indicates, these compounds break down proteins, are enzymes that break down inhibit enzymes that break down the proteins in the insect, and thus they affect how they digest their food. For example, I found that when caterpillars feed on tomato plants respond by increasing the amounts of proteinase inhibitors by threefold compared to undamaged plants. And this increase in the levels of proteinase inhibitors it correlates with a decrease in caterpillar performance. So caterpillars do worse on plants that have been induced by other caterpillars compared to undamaged plants. But not all herbivores induce the same response in tomato plants. So for example, feeding by the sucking herbivore, the potato aphid on tomatoes does not cause a change in proteinase inhibitor activity. And instead, when you have these aphids, these potato aphids on tomatoes, they increase susceptibility of the plants to caterpillars. So the caterpillars perform better on plants that are being damaged by aphids. So in addition, caterpillars, when they feed on a plant, they are known to induce the production of volatiles in the plants. And for example, here I show plants, cotton plants that have been either damaged by a caterpillar, the same herbivore that I mentioned before, Spodoptera exigua, the beet armyworm, and plants that have been damaged. And this is the amount of these different volatiles that are emitted by plants, either when they are damaged by caterpillars or undamaged. And you can see that caterpillar damage induces an increase in volatile emissions. So early on, I tested whether this volatile response in cotton plants was specific to the type of herbivore that feeds on them. So for these, I compared the volatile emissions from cotton plants when they were damaged by 3 herbivores that have different feeding habits. So I used chewing caterpillar, the beet armyworm, Spodoptera exigua. I used a cell content feeder. This is a piercing sucking herbivore, Lygus hesperus, and a phloem feeder, Bemisia tabaci. So the first two, the chewing caterpillar and the cell content feeder, they can cause a great amount of feeding damage on the plants, while the phloem feeder, when it feeds, is more gentle to the plant, cause less mechanical damage. However, they they are responsible for the transmission of a lot of diseases. So I found that the amount of volatiles emitted from cotton plants varies depending on the type of herbivore feeding on the plant and the type of feeding habit. So both the chewing caterpillar and the cell content feeder induce greater amounts of volatiles compared to the phloem feeder. So you can see that I found that the plants respond differently when they are damaged either by a single herbivore compared to when they were damaged by two herbivores. In this case, a caterpillar and whitefly nymphs. So you can see that when they are attacked by a single herbivore, by the caterpillar, the plants induce more volatiles compared to when they were damaged by both herbivores, the caterpillars and the nymphs. So the presence of the nymphs activate some defenses that reduce the response of the plants to the caterpillars. So you can see at least 3 compounds were emitted in lower quantities when the plants were damaged by both herbivores compared to the caterpillars alone. And this is important because caterpillars and aphids and other sucking herbivores induce different defensive pathways in plants. So on one hand, you have caterpillars that induce the jasmonic acid pathway that triggers the induction of defenses, such as what I mentioned, the proteinase inhibitors, but also they induce the induction of volatile production, so indirect defenses. But in contrast, aphids and other sucking insects, they induce, like whiteflies, they induce the salicylic acid pathway. And it's known that the activation of this salicylic acid pathway inhibits the jasmonic acid pathway. So you have this conflict when you have more than one herbivore feeding on a plant, on how the plant responds to them. So my studies were the first ones to document that multiple herbivores can influence volatile emissions in plants. I also showed in subsequent studies that the number and identity of herbivore species on plants can influence tritrophic interactions. And that's the work that you read. Also, that's some of the work that we conducted with Dr. Musser, where we looked at the plant response when they are damaged either by a single herbivore or damaged by two herbivores at the same time. And I also conducted a study that showed that the phenotype of the neighboring plant, whether they are induced or not induced, can affect the number of herbivores and natural enemies on the plants. So, so the identity, the phenotype of your neighbor has an effect on whether you get more or less herbivores and also natural enemies. So this brings me to my more recent research to manipulate natural enemies in agroecosystems using plant volatiles. So due to stricter regulatory restrictions on chemical control, there is an increased interest in ecologically based strategies to manage insect pests. And it is well known that crop diversity can promote biological control in agroecosystems by providing food and shelter to natural enemies. But this can be dependent on the composition of the surrounding landscape. So that's, it is believed that increasing diversity within and around the fields can increase ecosystem services that are provided by the natural enemies. So an expectation with that is that the natural enemies will move from the surrounded habitats to the focal crop to reduce the pest populations. But this might not always be the case. So to assist with this process, we could use synthetic volatiles that could be placed within the crop to attract natural enemies from these natural or semi-natural habitats. So for this, my lab has been studying whether we can manipulate natural enemy behavior using synthetic plant volatiles in agroecosystems. So let's talk about the work that we have done on this. So as I mentioned previously, plants increase volatile production and emission after herbivore feeding. So this response has been referred to as herbivore-induced plant volatiles. And these HIPVs play an important role in attracting the natural enemies of the herbivores. So they attract predators and parasitoids of the herbivores in agroecosystems. So these HIPVs are important in attracting natural enemies. It has been shown also in nature. For example, in a tritrophic interaction that consisted of wild tobacco plants, the tobacco hornworm, and the big eye bugs, Kessler and Baldwin found lower larval survival when plants were treated with methyl jasmonate. So you can see here, methyl jasmonate here, or when they were treated with different HIPVs. So when they were treated with cis-3-hexanol, linalool, and cis-alpha bergamotine. However, an open question remains on whether these HIPVs can be used to attract natural enemies to enhance biocontrol in agroecosystems. So for this to happen, three criteria must be met. So the first is that these HIPVs need to attract the natural enemies. So the second criteria is that this attraction should reduce the pest population. And the third criteria is that this reduction in pest population should cascade to reduce damage of the crop and also increase crop yield. So we looked at several studies that have tested these criteria. And so we found that a total of 43 studies that have actually shown that these HIPVs can attract natural enemies, so supporting the criteria number 1. However, fewer studies have actually looked at whether this attraction results in pest suppression. So only 13 out of those 43 have looked at whether there's any effect on the pest. And even fewer, only 3 of them have looked at whether this cascades down to affect the crop yield. So one HIPV that has received great attention in these studies is methyl salicylate. So methyl salicylate is emitted by the leaves and the flowers of many plant species. And it is commonly induced by insects with that have different feeding habits, such as spider mites, aphids, and the Colorado potato beetle. And can induce, this methyl salicylate can induce volatile emissions from the neighboring plants. It is also commercially available as a slow release dispenser called Predalure. So you can see here, to attract natural enemies. So you can actually buy this product and place it in your garden to attract natural enemies of the herbivores. So in a collaboration with Ian Kaplan from Purdue University, we conducted a meta-analysis to evaluate the magnitude of natural enemy response to methyl salicylate in the field. And we collected a total of 18 experiments in 14 publications that resulted in 91 observations. And in general, what we found from this meta-analysis is that natural enemies are attracted to methyl salicylate. So there was a positive response of natural enemies to methyl salicylate. And we did not find any differences between predators and parasitic Hymenoptera, or differences in the response across different taxa of the natural enemies. So in general, it seems like natural enemies of different groups of natural enemies do respond to methyl salicylate, are attracted to methyl salicylate. So in an early study, we found that lady beetles, lacewings, and syrphids are attracted to Predalure in cranberries. But this study was conducted in a relatively short amount of time. So it was only done in 1 year and in 1.5 months. So we wanted to look at whether these HIPBs provide long-term predator attraction, which is our criteria number 1. We also asked whether these HIPBs can increase ecosystem function, so whether they have an effect on the herbivore, and also whether they have an effect on ecosystem services, so whether they increase the yield or reduce the amount of damage in the crop. So to address the first criteria, we tested if the attraction of natural enemies to Predalure was reliable over time. And for this, we conducted a study in cranberries where we had sticky traps. So you see like yellow sticky traps, either baited with a Predalure or unbaited. We had those 2 treatments within the same bed. And we then counted the number of predators on the traps throughout the season, and we did this for 2 years. So we found that syrphid flies were highly attracted to Predalure. You can see here the number of syrphids on the Predalure traps and the controls. So you can see, like, especially during bloom, when they were very active, the numbers of syrphids was higher on the Predalure traps, baited traps, compared to the control traps. And this was consistent over both years. We also found that lady beetles were attracted to the Predalure, but only in 1 of the sampling dates. And this response was not consistent throughout the years. And one of the possible reasons for these inconsistencies is that the species composition of lady beetles varied between the 2 years. So you can see the number of species that we found in 1 year was very different from, on year 1 was very different from year 2. So, but these results indicate that we can attract natural enemies, especially syrphids in cranberries using these HIPBs like methyl salicylate. However, it's important to know if this attraction would lead to increases in predation, which is was our criteria number 2. So to look at that, we did a study where we placed sentinel egg masses. So you can see here these little wood sticks, and we would put like an egg mass there. We would glue the egg mass. These are European corn borer egg masses. And we would leave those eggs for 2 days in the field. So we would count the number of eggs before and after and calculate the difference. And this was done 6 to 8 times over the season and was repeated for 2 years. So in the first year, we found higher predation of eggs that were near the Predalure, especially later in the season. And this pattern continued the following year. And if we look at the total numbers, you can see that the amount of predation was higher when the eggs were near the predalure compared to the eggs that were near to the control. So with without the bait. And this was consistent for the 2 years. So the amount of predation was higher, in both years when they were placed near the predalure. So to observe who was responsible for this difference in predation, we set up cameras in cranberry beds where we recorded the predators visiting the sentinel egg masses when they were near the predalure. So we had these stations where we had the predalure, we had the egg masses, and we had a camera. So you can see the egg masses there. And we replicated several times during the season. So we did recordings in 8 different days for a total of 576 hours. So we went through the video recordings and we found that there were a total of 75 predator visits, and most of the visits were done by lady beetles. So more than 50% of them. And most of the, there was a difference between the visits when the eggs were near the predalure compared to the control. So most of the time, like 3/4 of the time, the lady beetles were visiting the eggs when they were near the predalure. And you can see here in the video, the lady beetles and visiting the eggs. And this led to higher predation of the eggs. So when the eggs were near the predalure, we observed twice the amount of egg predation compared to when they were near the control. So our third criteria is whether this could lead to a reduction in damage or an increase in yield. So are they providing, can we increase ecosystem services in agroecosystems by using these HIPVs? So a concern that was raised by Ian Kaplan and others with the use of HIPVs is the potential of attracting natural enemies to areas where the prey or the host are absent, which could actually disrupt biocontrol. So a strategy that could prevent this negative effect is by combining these HIPVs with other strategies, such as combining them with companion plants. So in this case, the companion plants would provide a reward to the natural enemies in the form of nectar or pollen. So this strategy has been called attract and reward. So you combine the use of HIPVs, for example, here to attract the natural enemies with a companion plant nearby to provide them with some food in case the predator, the host or the prey are absent in the crop. So we tested this concept in a system that consisted of bean plants and coriander as the companion plant, and methyl salicylate as the HIPV. We then measured the attraction of natural enemies and the effect of the natural enemies on the herbivores and on the amount of damage that they cause to the crop. And also we looked at the yield. So we measure ecosystem structure of the natural enemies, ecosystem function by looking at the impact of the natural enemies on the herbivores and how it cascaded to affect the plant. So we looked at the ecosystem services provided by the natural enemies. So these studies were done by my former PhD student, Giordano Salamanca in Brazil, where he created 7 replicated blocks of bean plants. And this study was done in 2 consecutive years. So in each block, he had 4 treatments. So one of the treatments was the control. So there was no companion plants or HIPV. One had the coriander alone, so only the companion plant. One had the methyl salicylate only, and the other one was the attract and reward treatment, where you had both the attractant and the companion plants. So for each of the treatments and blocks, he looked at the number of predators that were visiting the bean plants. He did that visually. So he just went to these locations, and he just counted the number of natural enemies that were on the bean plants. So he found a greater number of lady beetles in the bean plants with the coriander than in the other treatments. He also found more syrphids in all the manipulated treatments compared to the control. And here he found an additive effect of both the coriander and the HIPV. So if you add these two, you get this amount, similar to that amount. And also the predatory stink bugs were also higher in all the manipulated treatments. And there was no difference among the treatments. So he also measured the number of herbivores on the bean plants, as well as he also looked at the level of predation of aphids that were placed in these cardboards. So these were frozen aphids that were glued on the cardboard, and then he counted the number of aphids left after 24 hours. So he found lower amounts of spider mites in all the manipulated plots, but there was no difference on the number of herbivorous pentatomids and the number of chrysomelids. And he also found that predation of the aphids on the cardboards was higher in the attract and reward plot. So the plot that had both the coriander and the HIPV. But we were mainly interested also was like whether there was any effect on the plant itself. So any effect on yield and damage. So Giordano for this tested our criteria number 3 by measuring crop damage by counting the number of damaged leaves per plot. And also he measured yield by measuring the crop biomass per plot. So he counted the number of and the mass of each of the pods and the number of seeds that were present in each of the pods. And again, he measured the crop biomass also. So he dried the plants and then measured the biomass. So as far as crop damage, we found a lower damage in the leaves for all manipulative treatments, most likely due to the lower number of spider mites on those treatments. However, this did not translate to an increase in crop biomass. Also, we found no effects on the number. These are the total number of pods and seeds. So there was no effect on the total number of pods or the number of seeds or the weights of the pods and the seeds. So in conclusion, we showed a support of our criteria number 1, since the HIPVs enhanced ecosystem structure by attracting natural enemies either when the HIPV was alone or in combination of the companion plants. Our data also provided some support for criteria number 2, since the HIPVs can increase the ecosystem function by increasing predation rates, as we saw with the number of aphids that were consumed. However, our data only partially supported criteria number 3. Since HIPVs alone or in combination with companion plants, they reduce herbivore damage, likely because there was a suppression in spider mite populations, but did not result in an increase in crop yield. So it's becoming clear that if you want to use these HIPVs to attract natural enemies, we need to find methods to provide the natural enemies with food resources in case of when the host or the prey are absent. So this leads me to like the second part of my talk. So companion plants can provide supplementary food sources for natural enemies, such as pollen, extrafloral nectar, and also floral nectars. But in addition, plants also produce these droplets known as guttation. So you can see these small drops are along the edges of the leaf. And these are secretions along the edges that were thought to be water sources for insects. So you can see a close-up of the droplets. And this is fairly common in many plants. However, these guttation fluids are not just coming from the xylem, but also they are coming from the phloem sap and could be composed of more than just water. And this is work that was done by a former postdoc, Pablo Urbaneja Bernat. And what he found was that in blueberry fields, the guttation droplets were present throughout the season. So you can see like these are the different hours during the day, and these are the different times of the season. So you can see that the droplets were found throughout the season, but it peaked around June when the plant was putting new growth. And also there were higher numbers during midday. He also counted the number of insects that were visiting these droplets in the field. So, so these are insect taxa that he observed visiting the guttation droplets. And he found that Drosophilids, like flies, were visiting those droplets. Also, he found several like predators, like lacewings, chrysopids visiting the droplets. And also he found parasitoids feeding on them. So, so we asked whether this guttation increases the fitness of the insect. So we tested this using a herbivore. As I mentioned, there were a lot of flies visiting these droplets, so we used Drosophila susukii as a herbivore. We also look at a parasitoid, Aphidius ervi, and also a predator, as we saw lacewings feeding on these droplets. So we conducted experiments where we looked at the effect of guttation on the longevity of these insects. So we used newly emerged adults, and we fed them different diets. So we either fed them water alone, we fed them the guttation droplets, we fed them sugar, we fed them protein, or we fed them a combination of sugar and protein. So these diets were placed on paraffin pieces, as you can see here. And then we put the insect on a vial, and then each day we gave them we gave them a piece of paraffin with the diet. So what we found was the females of the flies live longer with the guttation. So the guttation is the color green here. And you can see when they are feeding on the guttation, they feed as long as when they were fed with the sugar diet. They live less when they were fed the protein or the sugar and the protein. And this, we found the same effect for the males. You can see that the guttation, when they were feeding on the guttation and the sugar diets, they live longer. For the parasitoid, the females live longer. For the parasitoid, you can see like also the same. So this is the guttation in green, and also the females live longer on the guttation and the sugar diet, and same for the males. They live longer with the guttation and the sugar diet. With the lacewing adults, you can see also the guttation and the sugar diet, and in this case also the sugar with the protein. They live longer, the females, and same for the males. We were also interested in the effect on fecundity. So we looked at egg load, the number of egg load. So we provided females the same diets. These were newly mated females that were fed the diets, and then we dissected the females 1 day after, 3 days after, and 7 days after feeding on the diets. So for the flies, we saw some differences after 1 day. You can see this is the sugar protein, and also with the guttation, we start to see separation after 1 day of feeding on those diets. By day 3, the guttation starts to separate even further. Also, you have a benefit from feeding on the protein and the sugar and the protein. By day 7, you can see that they are benefiting from the sugar and the protein, the guttation, and less from the sugar alone. For the parasitoid, you also see some separation. On day 1, you can see that the egg load was higher on the guttation by day 1. This is the same effect on day 3. The guttation, those that were fed guttation had more eggs than the rest, and also those that were fed the sugar and the protein. And they were separated also with the guttation and the sugar and the protein by day 7. For the lacewing, you see also the same effect. By day 1, there were more eggs on the sugar and protein and the guttation. Same for the day 3, the guttation started to have a stronger effect. Also the sugar and the protein, and same by day 7. You can see the guttation and the guttation here and the sugar and the protein increasing the level of the numbers of eggs in the females. So in conclusion, these studies report for the first time that guttation is not, that guttation is a nutrient-rich source for insects, that is not just water, that it contains, we show that it contains sugars and proteins that are nutritious for insects belonging to different gills. And it can be more also, we show that it's present throughout the season and throughout the day. So it can be more reliable than for example, nectar and pollen. We also found that insects from different taxa and feeding guilds, as I mentioned, visit these glutation droplets. And when they feed on this glutation, they increase their survival and also egg load. So to conclude and to leave you with some take-home messages. So based on our studies and also by others, it is possible to use these synthetic HIPVs to manipulate natural enemies in agroecosystems. But they are unlikely to work as a standalone strategy. So additional strategies will likely be needed in combination with the HIPVs to conserve natural enemies and also enhance biocontrol, such as providing them with food sources with the use of companion plants in an attract and reward scenario. And these plants, these companion plants, can be rich in pollen, nectar, but as I mentioned, they can also be rich on glutation. So there's still many remaining questions. So we need to do more research to address some of these questions. For example, whether multiple strategies are better than one. As I mentioned, it's likely that this is the case, but we need more studies to prove that. We also need to know whether natural enemies respond to the response of natural enemies to these HIPVs are reliable across multiple cropping systems. So we are currently conducting, in collaboration with some colleagues, we're conducting a meta-analysis to look at all the cases that have been done, all the case studies that have been done to look at the effect of HIPVs on natural enemies in agroecosystems and see whether there's some common, some patterns that come out on how these natural enemies are responding to these HIPVs. I have a grad student that is looking at how landscape features influence these tritrophic interactions. So as I mentioned at the beginning of the talk, natural enemies, these semi-natural habitats provide a source of natural enemies that move into the crops. So, so maybe it is possible that these HIPVs work better when you have this when the crop is near these semi-natural or natural habitats. So we're trying to show whether that's the case. And at the end, the question remains of whether farmers are going to be willing to adopt these strategies to manipulate the natural enemies in their farms. So with that, I want to thank you. This is my lab during COVID. And you can find information. I think a lot of you have already searched me on the web. You can always visit my website and contact me whenever you need me for anything. So with that, I'll take any questions and we can talk more about what I do.
All right, so I know that don't rush to jump ahead, but if you want to ask a question, it'd be great. Then it might take a second to formulate one.
I've actually got a question.
Good, Ava.
Yeah, so I was curious how you collect the glutation from the plants.
Oh, the blueberries have like a lot of gutations along the edges of the leaf, and they are fairly thick. So it's very like dense. Actually, you can touch them, and it's it has a lot of sugar on it. So what we just do is like we just grab them with a fine brush, and we just put them in a petri dish, and that's it. That's how we collect them. So
so they're solid?
Was that?
They're solid. I'm thinking more like sundews where they feel really like watery and viscous, but the ones on blueberry plants are solid. That's cool.
They're solid. Yes. Yes. Yeah, it seems like, and that's something that we are looking into now. Pablo is looking. He went back to Spain now. He's doing a postdoc there, but he's still working on glutation. And one of the things that that we got, one of the feedbacks that we got from reviewers when we submitted our paper was how common and how whether what we saw in blueberries actually happens in other crops, and whether the other crops can also provide this level of increasing fitness for natural enemies when they feed on it. And he's finding that actually we're also looking at the chemistry. Like I didn't show the chemistry. We have analyzed the levels of sugars and proteins in the glutation in blueberries. But if you look at across different crops, it's very variable. So not all the crops have this amount of sugar, and also not all the crops have the amount of protein. So it can be very variable. And for example, tomatoes don't seem to be as nutritious as the ones that we found in blueberries. But that's something that we are studying now. The early literature, most of the literature has been done with pollinators. If you look at literature on glutation and effect on insects, most of it is looking at how systemic insecticides can be present in, like neonicotinoids can be present in the glutation, and whether it has an effect on pollinators, but not necessarily on the positive effects of the glutation on the insects. And again, it was because mainly it was thought that it was just water, and you know, if you treat the plant with a systemic insecticide, that the plant will you know will have those insecticides will accumulate in the glutation, but not necessarily having a positive benefit on the insect.
Very cool. Thank you.
Yeah, that was what I was kind of wondering a little bit. So what's it called, degustation or
glutation?
It hasn't really been heavily characterized as far as what it is chemically. It's other than we know it's some sugars and proteins. There's what's the type of protein that you used in your mix, and how much similarity would there be?
So you can tell that it's not the same because it didn't provide the same effect. It was just yeast. So
Okay.
So that was the form of protein that we used, and sugar was like sucrose.
It seemed like it was helpful when the eggs were being developed anyway. I'm guessing there might be some more like free amino acids and things that would be easier to digest too, I'm guessing. And then you mentioned the plant with the methyl salicylate. What was the plant again? Coriander or something like that you said?
Yeah, coriander with the bean plants.
Yeah, so the coriander does it have the glutation, I mean?
That one we don't know. At that point, we didn't look at the glutation. We were definitely, like, the coriander has, you know, the pollen and possibly nectar that provides to the natural enemies. And that's what the natural enemies might have been more attracted to, especially like lady beetles. You could see that lady beetles were very attracted to it, also syrphids. And we find like syrphids are very attracted to like flowers. Like in cranberries also, syrphids are very attracted to the flowers of the cranberry flowers, most likely because they are looking for some source of food. And methyl salicylate is known to be emitted by flowers. So it could be that that they are queuing on a common volatile from flowers, and to find, some source of food for, when they are adults. But yes, like,
is the coriander a natural like cover crop? I wasn't sure why you used that one. I might have missed that, or eating a slice of pizza at the time.
yeah, it's commonly used for attracting as a companion plant to attract natural enemies.
Okay.
Yeah, buckwheat, coriander. Yeah, those are those are commonly.
They don't function as a weed though to the bean plants that any come out.
So it could be competing with the plant. It didn't seem like it, because when we looked at the yield, it didn't seem like to have any effect on the actual growth of the plant. So the purpose of it was just to find some plant that was attractive to natural enemies. So we used something that was commonly reported to attract natural enemies, and coriander is one of them. Dill is another one. Buckwheat is another one. So those are things that are commonly tested to attract natural enemies. And also was because my Giordano, the student that did the study, he did his master's looking at whether coriander attracted chrysopids and the effect on aphids. So he was looking at like coriander as a companion plant for chrysopids, for lacewings. So he found that it was attracting lacewings. So that's what's he used something that he was familiar with as a reward.
I don't want to hog things. I would ask you, what is this? What is a syrphid eat? It's been a long time since I've taken entomology.
Oh, hoverflies.
Okay.
Yeah, hoverflies. So the adults, they just feed on pollen, nectar. The larvae are predaceous. So the larvae prey on aphids.
That makes more sense to me. Did you have a question? You can come down if you want to.
Yeah, thank you so much for the presentation. So I was about to ask this, like for a farmer to adopt this type of method, the aim of or the purpose of farming is to make profit for a farmer. So like I can see some, maybe like, do you ever find out some kind of competition between the soybean and the coriander plant, which is the companion plant? Is there any kind of competition that might affect the yield of this soybean plant?
So, I mean, that's a good point. Like whether, so I think I know where the student is heading to, like whether a farmer will be willing like to use like land to plant a companion plant. And that's something that, depends on, I feel like it's going to depend on what benefits the farmer is going to get out from these companion plantings. And also like how you arrange it is going to be important. Like, whether it's an annual plant or a perennial plant, where you put these companion plants is going to vary, But one of the things that, and we wrote an annual review in entomology, looking at how, we could conserve natural enemies in this way. And one of the things that we conclude in that paper is that it is important, like, that these companion plants provide multiple services to the farmer. So one service can be in the as biological control, for example, as I talked, but also pollination would be important. So if you have some companion plants that not only attract natural enemies, but also attract pollinators, that would be ideal, likely for a farmer, because you're getting more services from those plantings. And it's more likely that a farmer is going to see an effect on the crop. Because if you attract pollinators and natural enemies, especially pollinators, you're going to see hopefully an increasing yield. So that's something that, you know, moving forward, we need to think about, you know, having these multiple benefits from having these companion plants. That was something that there's one study that I know of that tested that, and they found some impact on the crop in blueberries. But there's not many studies that have looked at that, because usually, like, the pollination people work on pollinators and the biocontrol people work on natural enemies, and there's usually, like, they don't do both. My student is doing both, so we are looking at both types of ecosystem services, you know. So I think we need more studies on that.
Yeah, thank you so much. I was kind of wondering, because I'm currently gathering some materials. I'm working on the effect of like fertilizer. When you fertilize caterpillar, your soybean plants. So when a caterpillar feeds on it, I'm trying to look at the effect. So I'm kind of want to correlate it with this, the companion plants. Now, our soybean plants, we are going to fertilize it. And there are some rates of fertilizer that we can apply onto our soybean plant to have a particular yield. So now, with that fertilizer rate, won't it affect again the companion plant? Like, what would be the correlation? Do you kind of test effect of fertilizer on those companion plants and the soybean plant?
So that's an interesting question. I thought you were going somewhere in a different way. Like, I like your ideas, and we just submitted an opinion piece to PNAS. I don't know if it's going to be accepted, but it looks at multiple strategies to control pests. And what we are arguing in that piece is that we should look into manipulating bottom-up and top-down effects. So so the bottom-up would be, like, for example, fertilizer, like what you're saying, that can change, you know, the nutritional composition of the plant, but also, you know, manipulating the top-down, which are the natural enemies. So so I like your idea. Like, you know, if you look at the effect of fertilizer or any, like, there's a lot of studies like now with on soil health. So, like, manipulating things in the soil that make the plant maybe more resistant to the insect, but at the same time, like, doing some strategies that can manipulate natural enemies. So the strategies can be compatible. So you can have the plant producing defenses, and also might be compatible with biological control. So if you have, like, you know, if you change the soil composition, if you have, you know, something that makes the plant stronger with some type of fertilizer, and also you have a companion plant, you can manipulate those two, you know, and hopefully you get better suppression of the pest.
Thank you so much.
but that's a very, very interesting. Yes, I'm actually very interested in that question myself. We're looking at these bottom-up, top-down effects and how we can manipulate them and effects on these trade traffic interactions. So so we're doing some. I have a postdoc, a PhD student working on soil health. So we're looking at different types of soils in blueberries. We're categorizing the different soil types, and we're looking at the growth of the plant on those types of soils and how it affects the defenses in the plant and the herbivores that feed on the plants.
More questions in the audience? I thought I was going to ask you something else, but so the methyl salicylate, does that make the plants more susceptible to chewing insects?
So it could be. We haven't looked at that. I mean, we're testing it just as a bait for like natural enemies. But it could be that some of the effects that we saw, for example, with the spider mites, it could be through some kind of induced responses of the plant and activation of some kind of defenses. So we didn't see any caterpillars, like increasing caterpillar damage, for example, on the plants. So as you saw, like there was no caterpillars. It was like spider mites and some other piercing, sucking. Well, the chewing, there was a chrysomelid, but we did not see, like in the chrysomelid, we didn't see an increase in chrysomelid damage. I mean, there was a trend, which was kind of interesting, but there was not significant. So it could be that you could make the plant more susceptible to some herbivores, but we did not directly test that idea. But that's I get that question a lot. Yeah.
Anybody else have a question online or in person? All right, so we give it a little round of applause again.
You said you wanted me to talk about my position.
Well, or developing a scientific career, maybe to spend 5 or 10 minutes just talking a little bit about your journey, I guess you could say, or what you're doing now. Because obviously, I mentioned that you have that dual role where you're helping growers in your community as well as having a research program. I don't know if you teach a formal class or two, but I know you're working with growers extensively.
I actually didn't realize that we were already like it's 5:30 for you. Yeah, so I do a lot of extension. So I work closely with growers. So 70% of my appointment is in extension. So so everything that I do has to be kind of translational into some practices that the growers can use. So things that we do that are even basic, we try to like look into how, like, mechanisms and try to see whether we can manipulate things in the field to reduce a pest. So so with my job, I do a lot of talks to growers. I organize meetings. I write blogs. I write newsletter articles. We do field demonstrations, things like that. I also work with, a lot of the work that we do is on farm. So we constantly talk to growers. We're in constant communication with growers. Tomorrow, for example, I'm going to go to a farm to monitor for insects. So we do a lot of this type of work where we do a lot of the work that we do is on farm. So we visit a lot of farms, which I enjoy because I feel like I never trained as an extension specialist. You know, I didn't know what an extension specialist was when I was a grad student. I don't think we were. Nobody mentioned much about extension when we were in grad school. I think students nowadays are more exposed to extension because I think there's a need to know what extension is because a lot of positions need extension. So I see that a lot of students, and I have all my students participate in extension. You know, they all come to grower meetings. They all come to, they see me give talks to the growers, how I interact. You know, they visit farms, they talk to the farmers, so they get that exposure and see what, you know, what my work is about. But I had no, as you could see with my background, I had no experience with extension. I was a researcher, and I was surprised when I got this job. But I enjoy it because, you know, I feel like you are making a difference by working with growers and trying to change their behavior. You know.
Yeah, that sounds great. Anything else? Well, I think we really appreciate your visit. I think we learned a lot, actually. So,
thank you. Thank you,
Thank you so much. Yep. Thank you so much.
Thank you for inviting me.
Yeah, my pleasure. It was like all ours. Yeah. Yep. Ava says thank you too, and I know everybody seems pretty appreciative.
I think she left already.
All righty, sir. Well, we appreciate it. Well,
thank you for the invitation.
Take care.
Okay. See you. Hopefully, I'll see you at ESA. Are you coming?
Is it too late to sign up?
Actually, the abstracts are due in 11 days on May 15th.
And where's it at this year?
It's in Maryland. It's close to here.
Oh, all right. Well, you take care.
Okay. Hopefully, I'll see you then.
Sounds good. Thanks for that info. Thank you.
Okay. Bye-bye.